So this is the part 3, chapter 4, section 4.3, and we're going to discuss electron microscopy in this chapter. So far, the discussion have been the light microscopy and the variation of the light microscopy, and our beam of radiation was light. Now I want to switch to the electron microscopy, where the beam of radiation is electrons and where the wavelength of the electrons is substantially smaller than the wavelength of light. It produces a spectacular resolution with the electron microscope. So let's calculate the resolution of a typical electron microscope using this formula that we have learned previously. So let's calculate the resolution of a typical electron microscope using this. So to do this, let's first take a look that shortest lambda, which is a beam of light, is about 0.45 micron, 450 nanometer, and D is 2 micrometer. For the electron microscope, the shortest beam of electrons is about 0.005 nanometer. And the numerical aperture that is the best possible microscope can produce, the n sin alpha right here, the numerical aperture is about 0.0305. That's limited by the technology. So if we just plug these numbers, 0.61 multiplied by this number, and if we just over 0.0305, you will find that the D is approximately, or D, or the resolution, is about 0.1 nanometer, which is about 2,000 times better than the light microscope. So you can see that there is a huge improvement over the light microscope using the electron microscope. So here, this is not required for your quiz and exam, just giving an idea that what we mean by the spatial and temporal resolution and where our techniques actually do stand in all of the imaging techniques that is available. So here we are looking at, this is a micrometer range to about nanometer range, 1 nanometer to approximately 0.1 angstrom. So remember that 1 angstrom is 10 nanometer. On the other hand, this is the scale of femtosecond, picosecond, nanosecond, microsecond, millisecond, second to kilosecond. This is what you are seeing, the temporal resolution. So this actually calculates techniques which are used to calculate any dynamic process is on this scale. And this is the resolution. The separation of 2 points resolution is the spatial resolution. So this is actually pretty much that what we actually covered. So think about that we talked about the confocal, turf, the single molecule tracking, super resolution fluorescence, and we're going to discuss the cryo-TEM, the transmission electron microscopy, single particle cryo-EM. These are the things that we are going to discuss. There are many other different types of techniques that is actually being used for this purpose, but we are just limited to in here. And you can see that there are single molecule or particle cryo-TEM that can actually go to the atomic resolution of 0.1 angstrom and close to the X-ray crystallography and other techniques, which were actually much more advantageous for many, many years, although they're very difficult techniques. But you can see that electron microscopy actually can almost compete with all of these techniques. So here we are actually just trying to give an overview of the comparison of the light transmission and the scanning electron microscopy. And you can see that light microscopy do have a light source, and then you have a condenser lens, and the light which is coming out of the light source is actually focused onto the specimen. From there, it goes to the objective lens, then the eyepiece or the projection lens, and this is the vision that you actually can see. On the other hand, in the transmission electron microscope, the electron or the scanning electron microscope, the source of the electron is actually a tungsten coil, and a huge amount of voltage is actually applied across that. So usually it starts about 50 to 70 kilovolt, the same for the scanning electron microscopy, but the voltage of that tungsten coil can, for cryo-EM's state of the art, can go up to 300 kilovolt. So this huge amount of power that is given to this tungsten coil, there's a stream of electron ejected at that point, a stream of electron. When these stream of electron actually eject from there, there are some same way that you have some condenser lenses, there are some objective lenses and projection lenses. What they do, that these are actually electromagnetic lenses. They are just electromagnetic rings, and you can actually adjust them to focus the electron beam exactly like the glass lenses that we use in the light microscopy. So here you focus the beam onto the specimen, it goes through the objective lens, then to the projection lens, and then it goes onto a fluorescent screen. So this is the most important part of the transmission electron microscope, that with your eye, you cannot visualize an electron because it's completely invisible. But if you have a fluorescent screen, commonly also known as a phosphor screen, what happens that at the base, if you have this, and then from here, you can actually visualize, there will be some eyepieces right here that you can visualize this screen. So what happens that when an electron actually hits that fluorescent or the phosphor screen, it will actually produce the light. So this is actually the technique, the same, if you remember, that your age-old televisions that we used to use, these box TVs, actually acted some way like this. You actually do have an electron generator, the cathode ray tube, and these electrons will hit the phosphor screen, which is the front of the screen of the television, and a bright spot will be generated. So this is the same way that an electron microscope actually works, and you can take images of with a high resolution camera, you can take the pictures of that as well. So the scanning electron micrograph, on the other hand, it varies a little bit, where you actually take almost like a three-dimensional image that we're going to talk about. But here you actually scan the surface of the image or the surface of the sample with a scanning coil, and this is called a beam deflector. So this will actually move the beam this back and forth, x-axis and the y-axis. This is a sample, 3D sample, and this is usually metal coated. Electrons will fall upon them and then they will be deflected, and these deflected electrons will be actually received by the detector, which actually will go to the camera and a computer monitor. And in all different directions, you can actually see this. So what happens that when you do scan the surface of that, you actually get a three-dimensional image of the surface as well. So the difference is that here you have to have a very thin section or a very tiny particle like a virus to observe that. So here the principle is some of the electron beams will pass through the specimen, some of them will not. That will generate the contrast in the electron microscope. Again, remember that this is the principle, whereas in the scanning electron microscope, no electron passes through the sample because the sample is coated by a heavy metal, and all the electrons are deflected depending upon that what is the typical topography of the sample. So what will happen that when the detector is actually detecting the beam, if the topography is deep, then the electron will take a little bit longer time to travel to the detector compared to a surface which will go fast, a little bit faster. So this is basically the technique that doing this scanning, you can get a three-dimensional image of the sample. So here we are looking at a typical TEM, TEM, and the SEM. These are getting smaller and smaller. And a typical right here, what you are seeing that right at the very top, you have the coil, tungsten coil, which is the electron source. Then you have the condenser right here that you're seeing. This is a side tube where this is a sample or the specimen holder. There's like a small tube opening as such, and everything is kept in vacuum. So this whole thing is under vacuum, and you actually take out that small rod and put it in there, and then you vacuum seal it. So automatically it will do that. So that's your sample. Then you have the objective projection lens. At the bottom of this right here, you actually have the fluorescent screen that you visualize with your eyepieces right here. And underneath there are cameras which will actually capture the images as well. So these are all your control for your condenser, then objective, as well as the projection lenses. On the other hand, here you are looking at a typical scanning electron microscope. In the scanning electron microscope, the sample holder is right here, and you have to put the sample in. Right here, you have the electron source, and usually it is much smaller because you need a less amount of voltage or electron beam from scanning electron microscopy. So here is the detector. You can see that this is exactly the same way. It is in an angle. So from here, the image is detected through the camera using this detector, and that will be visible onto the computer screen. So here we are looking at the sample preparation for TEM is actually a little bit difficult. So the main disadvantage of the transmission electron microscopy is it's a complicated sample preparation. The images are great, but it takes a long time to prepare the specimens. You use the same 3 steps. You use the fixation, embedding, sectioning, and staining. Fixation is carried out using the cross-linking agents such as light microscopy, same like a glutaraldehyde. But there's a second fixation compound usually used for TEM, and usually that is sometimes called osmium tetroxide. So that's also used as the negative staining reagent, which is written right here, but that is also sometimes used as a fixative agent along with the glutaraldehyde, which I did not mention right here because it takes care of that process in this. So it is very important because it binds and stabilizes the lipid bilayer, which is very important in TEM because when you section, you actually use much thinner layers than the light microscopy. But the osmium tetroxide is also electron dense and serves as a stain, which I will discuss shortly. The second step is the embedding, and the specimen must be sectioned much thinner than for the light microscopy. The penetrating power of the electron is much lower than the light, so that the layer that has to be much thinner as well. So the electron gets through the specimen and detected on the computer screen. The embedding is done by the dehydration of the sample, and the sample is encased in a solid plastic block, which is could be epoxy or a solid plastic. And this process is not required for the viruses. This is only for the thick sample. And then you use an ultramicrotome or a cryomicrotome where the sample is almost close to a liquid nitrogen temperature. And that actually make the section much smoother and much clearer. And the sections has to be in the range of 50 to 100 nanometer thick. So because the electrons have limited penetrating power and they will not be able to pass through them. So this is the overall process of the sample preparation for transmission electron micrograph. So the different organelles in the cell will pick up the stain in a more or less efficiently. The organelle that picking up the stain poorly will allow the electrons to pass through, and those electrons will reach the detector and appear very light right here. An organelle that absorbs the stain very efficiently will actually deflect the most of the electrons. The heavy metal stains are very dense, which we discussed right here. And these heavy metal stains could be uranyl acetate, the lead acetate, osmium tetroxide, which also works as a fixative after the glutaraldehyde as well. But again, if there is a cell with a lipid bilayer membrane is needed, then we use that in this step. Otherwise, we can just use this one as a negative stain. Phosphotungstic acid, and if you just look at that, these are the heavier, very heavy and extremely toxic, each one of them as well. And these are your stains for your electron microscopy. So right here, you can see, right, some of the organelles will not pick up the stain, electrons will pass through, and some of them will pick up the stain. And as these are heavy metals, the electron will be deflected by them, and you will see this as a dark spot. So the heavy metal stains are very dense, which means that it deflects the electron and very few electrons will get through to the detector, and that portion of the specimen will look very dark. So here we are looking at that how we actually prepare the sample. We first need some of these copper grids. This copper grids about 3 millimeter in diameter. And this is kind of, you can think that kind of microscopic glass slide for EM. So we have used glass slides with a cover slip for the most of our light microscopy, but here is a copper grid and it's tiny, very small. And what you do that you either buy or you can actually use a machine in your laboratory to do this. This has to be coated by a carbon or a formware, which is a form of plastic. So this is coated with a carbon, and immediately you will think that that means that this will be completely black or something like that. No, it is not. So the layer of carbon or formware is so thin that it will completely appear as transparent. It's almost sometimes difficult to see as a naked eye that where there is a film on that copper grid or not. So the reason for that why it is so thin layer of carbon or formware, because it has to allow the electrons to pass through it. And why these grids are there? Because remember that one small grid that you are seeing, that is a huge area for the electron microscopy. So this has nothing to do other than thus supporting that whole carbon or the formware film. Once those are there, you actually add the sample of top of them, and then you can actually stain directly because the samples will immediately attach by the electrostatics to the carbon and formware, and you can stain by the heavy metal, wash them, and now they are ready to be loaded onto the electron microscope. So here we are looking at a transmission electron microscopic image, and these cells are from the onion root, and you can see it in a spectacular detail. You can see the nucleus, you can see the nucleolus. This is the nucleus, nucleolus. You can see the plastid, you can see the cell wall, Golgi, vacuoles, mitochondria right here, endoplasmic reticulum, and you can see everything, amazing detail. So how do you get the contrast? For example, the dark nucleolus is in the nucleus versus the vesicles appear white. So the contrast depends upon two things. One of the type of macromolecules found in the various areas of the organelle of the cell, and the greater the atomic number of the macromolecules in that region of the cell, the greater the electron scattering, which produce a greater contrast. And these regions will actually appear as darker. So this is actually the principle that how it actually works. So right here, on the other hand, you are actually looking at some of the rotaviruses, purified sample directly loaded onto the grid. And you can see that this is approximately 100 nanometer in the width. So what happens that these dark areas are where you have stained, the stains are remaining in those areas, and the white areas that there is no stain and appear light. And that's how you can get that contrast. On the other hand, right here, you can see that here the cell tissue actually needed the fixing, where you can see that you have a mitochondria. Right here, you have an endoplasmic reticulum. And what is the difference in between that? That these are actually extremely small viruses, 100 nanometer in diameter. So you can directly actually put them on the copper grid, on the former film. Right here, this needs sectioning. This doesn't need section. And what you are observing, if this is a typical cell, you are only looking at the sample at just one section of the whole cell. So this is the plane of the section. So make sure that you understand this clearly, that when you are looking at an electron micrograph of a cell, it is just one plane section of the cell. So here you can actually looking at a neutrophil is internalizing two bacteria by the phagocytosis. And this is also an amazing detail that two bacteria that are internalized by this. These are the pseudopodia formed by the neutrophil, and you can appreciate how much detail you can see. So all these dark spots are the electron dense material, and you can see that the light areas are actually electron porous material. So here is another example. This is a dictyostelium. This is an amoeba that we actually talked about, and these actually picked up some of the latex beads by the endocytosis, which is being observed by the transmission electron micrograph. So we can actually construct three-dimensional images from the transmission electron micrograph. And the beauty of this is that the cells are actually carefully sectioned in a serial plane. So you have to keep track when you are actually sectioning that tissue sample so that they are one after another. And then you actually take the individual TEM images and record at each plane. And you can actually using the computer, actually reconstruct those single plane images into this multi-layer images, which will look like a three-dimensional image. So this is a Golgi in a three-dimensional reconstruction. What you are seeing that these are the white small vesicles. These are actually the transport vesicles, which actually arose from the endoplasmic reticulum, and they are actually coalescing, or they are fusing with this cis face of Golgi. So this is the receiving cis face of the Golgi, but you do not see them this is the trans face of Golgi. You do not see them on the trans face. So what's going to happen that once they actually arrive in the Golgi, all these different proteins will be sorted into particular complexes, and then the Golgi will produce its own vesicles that will actually come out from there, either distributed in the cell or go somewhere else. So amazing 3D images can be reconstructed from the transmission electron microscopy. So here is another technique which we call the transmission electron microscopy for the metal shadowing. So here you take a glass or a mica surface and you have a sample. And this could be a virus, this could be a cell. And what you do that, say for example, if it's a cells growing on a glass or mica cover slip, you can take that. So then you fix that sample, and then onto the sample, you actually add an evaporated platinum. And how you actually do this process, you have a chamber which is in a high vacuum, and there is a source of platinum, and it is actually heated with a huge amount of heat. And what happens that it actually sprays kind of a platinum into a very thin film onto the sample, very, very thin film, so that it actually exactly like the three-dimensional image of the sample. So this is what we call as a metal replica. So now what you do that just to keep this metal replica properly in place, we actually use evaporated carbon just on top of that as well. So this is actually just for reinforcing the metal film. Now what you do that you cannot put this glass or the mica surface, which is much, much thicker, onto the electron microscopic grid. You cannot just put that. This is much large where your sample was growing. So what you do that usually you use hydrofluoric acid. Hydrofluoric acid is extremely corrosive for glass or mica, and it will actually dissolve this glass. And then you just have this top part, which is a metal replica. And now you actually load this metal replica onto the copper grid and you visualize them. So here you can looking at the metal shadowing of some poliovirus. This is actually, on the other hand, looks almost close to a scanning electron micrograph, but this is actually a metal shadowing by the transmission electron micrograph, and we will compare and contrast that what is the difference between this technique and scanning electron micrograph. Usually the transmission electron micrograph, you actually do have a much, much bigger, greater resolution. That's the most important thing. Then we are looking at a cytoskeleton. You can see that how much greater detail you can look at the cytoskeleton. And this was also done by using this metal shadowing technique. You coat that with a platinum. Immunoelectron microscopy, on the other hand, is very similar to the microscopic technique that we have seen in light microscopy. This is the same way that if you have some of the organelles that pick up stain poorly, they are not electron dense. On the other hand, the organelles that pick up the stain efficiently will deflect the electrons. Now you have what you do that organelles, we actually stain them with an immunogold. There will be an antibody, and attached to that will be a gold particle. And these gold particles are actually electron dense, so they will actually deflect the electron the same way as the organelles which are coated with this electron dense material like a stain. Only few will reach there. So again, as these will deflect the electrons, they will not reach and they will appear as dark spots. So this is the principle of immunoelectron microscopy. So let's take a look at that. There were variations on the fluorescence microscopy known as immunofluorescence microscopy, which relies on the antibody that binds to your protein of interest. So there's a similar variation known as the immunoelectron microscopy, and it relies on an antibody that binds specifically to your protein of interest and a secondary antibody. But in EM, the secondary antibody is not connected to a fluorophore, but it's covalently connected to a gold particle. Because gold is very electron dense, it can deflect the electrons and make those regions of the cell very dark. So you can identify the specific proteins on a transmission EM. But there is a variation. If you do not have a gold labeled secondary antibody can be used, and that can bind the primary antibody. And sometimes what we actually do that you can obtain commercially available gold particles that are bound to the protein A. This protein A is isolated from the bacteria Staphylococcus aureus, and it specifically binds, this protein A specifically binds to the Fc portion of the antibody, the constant Fc portion that we talked about. And this is a second way to label your primary antibody for visualization. Remember that if you have a primary antibody, you can always use a secondary antibody attached with a gold. This is the technique. On the other hand, if you do not need a secondary antibody, you can use your primary antibody and then use your protein A attached gold to stain your sample as well. So here, in this example, this is an antibody that specifically binds to the catalase, which is found in the organelle known as peroxisomes. So how the gold particles work? As mentioned, the gold is very electron dense. So if the peroxisome is stained with the anti-catalase antibody and with the gold labeled protein A, then the gold particles that are bound to the organelle will deflect the electrons and will identify that organelle. or the proteins in that organelle. These are peroxisomes, and you can see the tiny black dots, which are individual gold particles. Peroxisomes actually have very clearly. So here in this picture, what you are seeing, that this is for a HeLa cell, and this is actually an HIV. There is another one right here, but this is HIV is actually budding off from a HeLa cell. It's still attached with its membrane. So the HIV actually have just two components right here that is being of interest right here, not going into the detail. It does have an envelope right here, which is derived from the membrane. So it's a membranous envelope. And then you have a capsid right here. So here we are doing a double antibody labeling where we have used the first primary antibody, and the primary antibody is against the capsid protein. So the primary antibody binds to the capsid protein. And then we have added protein A, added 5 nanometer gold particles, 5 nanometer gold, and they actually will go and they will bind to the target, which is the capsid protein. Now what we do that we actually wash this sample and we inactivate this protein A sites so that there is no non-specific binding. Now we actually add the second antibody, second primary antibody, which is against that envelope, which is the anti-envelope antibody. So this primary antibody will bind to that. Now we again add the protein A gold, but here there's a little difference. Instead of a 5 nanometer gold, here the gold particle size is 10 nanometer, just a little bit larger. And once the excess amount is washed away and the sample is actually visualized, you can clearly see that the capsid actually do contain these a little bit smaller dots compared to the larger one right here. Because these larger dots are against the envelope, which is outside, and these are 10 nanometer gold particle, and these are 5 nanometer gold particle. So just by varying these gold particles, you can actually differentiate between the two different types. And this is a very powerful technique so that you can actually do a double, triple labeling to in the same electron micrograph, you can identify more than one protein where the location is or where is the localization. So here we are looking at a kind of a modification of the cryo-electron microscopy, and these setups are like amazing setups. I mean, like at the low end, that's probably cost you something like a $5 million, to the upper end, $15 to $20 million. We probably do not have any one of them in South Carolina. The closest is probably at the Duke Medical Center in North Carolina that we can think about. And it's a very state-of-art technology. So you can see that this is a Titan Kraus from the FEI. And if you just open these doors, it is like almost like a science fiction inside. It's a very, very sophisticated machine. But basically, what it is all about, that it is a very powerful transmission electron microscope. First of all, it uses an extremely high voltage for the electron to come out of the tungsten. So you can easily see that. So very powerful or bright. And the sample prep and imaging technique is different. So what we actually do in this cryo-electron microscopy, why we call this cryo, because we actually take the sample just in water. We do not need to fix it. We just do not need to dehydrate it, or we do not need to do anything. All we have to do that we actually take this sample in water and we actually freeze that quickly, that water in liquid nitrogen. When you freeze that water rapidly in a very low temperature like liquid nitrogen, it forms a vitreous ice. What is mean by vitreous ice? That this is actually almost like a glass, and there will be no crystal formation. So if there is an ice crystal formation in the cell, it will drastically damage the cell, which you call as an amorphous ice crystals. But there will be nothing like that. It will be frozen in a complete glass-like fixative, which you like a epoxy block or the plastic embedding media that we have used. And just the ice will transform into that kind of material by this quick freezing in liquid nitrogen. Then we actually load the sample onto the specimen, and this is constantly near the liquid nitrogen temperature, and it is kept in a high vacuum. Remember that the chamber where the sample or the electron beams are passing needed to be in a high vacuum because any air will deflect the beams. And also this sample needed to be at the liquid nitrogen temperature so that the ice remain in that vitreous ice condition. We try to keep that sample always at the close to the liquid nitrogen temperature. So even in the high vacuum, there is no evaporation of the vitreous ice. So once we actually put the sample on the stage is actually rotated at plus minus 70 degree in 1 or 2 degree increments in all different directions, in X and Y directions. So when these are tilted, the stages are tilted, each time the detected image is going to get just a slightly different angular view of the image. This is flat, just another angle, and you are going to get a lot of angle. So all these images are actually detected where same particle or image is going to be detected in different orientations. Now, all these collected images are actually done by the 3D computer. Reconstruction of all images gives you a 3-dimensional image. It's an extremely powerful algorithm, and nowadays the power of cryo-electron microscopy is at the atomic resolution, and it is a mind-boggling technique and state-of-art procedure. So here you can see that the cryo-electron tomography is basically, we were looking at this sample that this is a nuclear pore complex. Here, what you are seeing on the other hand, if you very carefully look at that, you can see these tiny nuclear complexes right here. These are the tiny nuclear complexes. You can see those. So here you can see the nuclear complexes. This is much more. On the other hand, this is a much more enhanced picture or much more magnification, where you can see that this is actually the picture of the nuclear pore complex, just one of them right here. So these pictures, many different ways that you can actually collect these pictures. And then if you reconstruct, this is the reconstruction from the previous picture right here. So from the previous picture, the reconstruction gave you each of these nuclear pores. From the reconstruction of the image from here, it actually gave you kind of, to some extent, the bottom portion of the nuclear pore. So you can see that the bottom portion of the nuclear pore right here. On the other hand, this actually gave the top portion of the nuclear pore. So you can imagine that this is like an unprecedented detailed structure of the ultramicroscopic 3-dimensional structure that can be achieved by cryo-electron tomography, which is why we call it a tomography, because it is basically looking at a 3-dimensional reconstruction, a 3-dimensional topography. That's why it's called a tomography. So you can appreciate right here, this is not required for your quiz exam, that a stunning 1.8 angstrom resolution cryo-electron density map. So what you are seeing that when you actually look at these images, you can actually have an electron density map. And if you look at this electron density map, you can actually look at that. If you just put an arginine, this is the molecule. So if you just look at that, this end of the arginine molecule is right over here, and it just goes on around this. So this right here is the oxygen is right here. So you can look at that. This is the phenylalanine. You can see the ring right here, and this is the rest of the molecule. On the other hand, this is a tyrosine, and you can see that the rest of the molecule is the same. Look at the beauty of that. You have right here the ring, but this ring actually have an hydroxyl right here. You can actually determine that hydroxyl right here. So this is your tyrosine, but this is phenylalanine. You do not have that tip or the hydroxyl right here. So you can imagine that how much molecular detail you can actually get from these cryo-electron microscopic references at ultramicroscopic structures. So the next one that we are going to talk about is the transmission EM requires an extremely thin sectioning and provide excellent 2D information and detail, but loses the 3D information from the specimen. But again, remember that we can do some techniques which we call as a metal shadowing, or in the cryo-electron micrography, we can actually do the 3D image construction as well. On the other hand, what happened that, remember that the confocal microscopy solved the problem with the fluorescence microscopy. So the solution of that problem for transmission electron microscopy is a different kind of electron microscopy known as a scanning electron microscopy. It does not require the sectioning of your specimen and will provide the 3D image and the information of three-dimensional characteristic of the sample. ACM, you can use whole cell or unsectioned tissues which are fixed, dry, dehydrated, and coated with a heavy metal such as a platinum, gold, something like that, which will deflect the electrons. So here is a comparison of the scanning EM with the transmission EM and Nomarski light microscopy. So you can right here see that these are ciliary projections from the hair cells of inner ear of a bullfrog. And in the upper right image right here, the Nomarski or the differential interference microscopy, you get some idea of the three-dimensional structure of this projection from their hair cells. Remember the resolution to be approximately 0.2 microns. The transmission EM on the panel below shows amazing detail, but really there is no idea about the three-dimensional nature of the structure. And here on the left, the image of the same structure on ACM, so you can sacrifice some of the detail in the ACM, but can obtain some excellent information about the 3D nature of the structure and the materials. So here on this other figure, that you can actually see that this is actually an unsectioned tissue sample of the lower respiratory tract. And you can appreciate that these are the three-dimensional structure of the cilia, which actually is towards the lumen or the empty space of your trachea or the tube. And these are the goblet cells, which actually secrete mucus. And these are your elongated epithelial cells, which are the columnar epithelial cells. And you can see their nucleus and the other structures as well. So amazing detail of three-dimensional structure that you can see with this scanning electron micrograph. So here is also an amazing detail. This is a scanning EM of a macrophage. They're also phagocytic cells, and in this picture, a macrophage is actually internalizing by phagocytosis the two red blood cells. And remember that the red blood cells have a finite lifetime, so after about 120 days, they will be actually engulfed and recycled by these macrophages. And the macrophage is a large hairy structure in the middle, and two red blood cells are the smooth structures. And you can see the smooth collar that is actually being formed right here. You can see that smooth collar right here. You can see that this is the smooth collar that is being formed, and that's forming around the RBC. This is actually the extension of the plasma membrane that is moving around the red blood cell, and that will engulf the whole red blood cell. So this is basically the pseudopodia formation. Amazing pictures that you can actually see through these microscopes. So that will be the end of part 3, which is a chapter 4.3, electron microscopy. The next part 4 will be the section 4.4, which is the isolation of the cell organelles. So far, the first 4 parts, we have seen that considering the techniques that focus on the whole cell. So now let's turn to techniques that focuses on the isolation and characterization of organelles and the macromolecules in the organelles. The first step in isolating organelles from the cell is to disrupt the plasma membrane. And in this case of a, say, it could be a plant cell, it could be an animal cell. So in a plant cell, you have to disrupt the plant cell wall. In an animal cell, the disruption does not require to, you just only need to disrupt the membrane or the plasma membrane. You do not have a cell wall. But no matter what, this disruption must be very gentle in order to preserve the structure and function of the organelles. So disruption is usually achieved by the presence of an osmoprotector. What is an osmoprotector? It's often the cells are disrupted in presence of, say, approximately 0.25 molar sucrose solution, chilled sucrose solution, which actually protect the integrity of the organelles when the cells are actually disrupted. There are a number of ways to disrupt the plasma membrane, such as ultrasonic vibration, or this is what we call is a sonication. This is actually using a very high sonic vibration to do this. We can use a tissue homogenizer, high-speed blender, and you can also force the cell through a filter with small pores, usually 8 micron. Usually, it disrupts the plasma membrane without damaging the organelle. So once you disrupt the cells, you have what is called an homogenate. Homogenate is sometimes called an extract as well. The homogenate contains a mixed population of particles of different density, shape, charge, and size. Because of the unique feature of all these particles within the homogenate, you can take advantage of that and use centrifugation to further purify the organelles. Because the organelles have the different density, shape, charge, and size, they have a very different sedimentation rate. This is why you can apply centrifugation as the second step. So the most common second step in organelle purification is differential velocity centrifugation. This diagram outlines the principle of this type of centrifugation. First, you filter your extract or homogenate to remove the clumps of cells or unbroken cells. You take the filtered homogenate and apply to a low speed spin for a short period of time. So in this diagram, you would centrifuge the filtered homogenate at 600 G for about 10 minutes. The resulting pellet contains the cell nuclei. So if you only collect that pellet, you can actually study the nuclei from the sample. But if you want to study other organelles, you can take the supernatant from that pellet, apply higher speed spins. In this example, 15,000 G for right here is a 15,000 G for 5 minutes under those conditions, and you will pellet mitochondria, chloroplast from plant cells, lysosomes, and peroxisomes as well. You can take the supernatant to a higher spins for longer spin, say for example 100,000 G for 60 minutes, and as soon as you just go to the 100,000 G, this becomes what is called an ultracentrifuge. This is turbine driven, complete vacuum, again state-of-art machines. You will pellet the portions of the plasma membrane and the endoplasmic reticulum and some other larger polyribosomes that may have been in the cytosol or attached to endoplasmic reticulum. So this is what you can actually get at that point. So further higher speed, you will actually end up with a supernatant that is fairly representative of ribosomal subunit or small polyribosomes, and you pour out the soluble part will be your cytosol. So now you have purified the organelles, maybe not pure enough. So let's focus on the pellet, which actually contains the mitochondria, chloroplast, lysosome, peroxisome, and see that whether we can be purified further. All of these organelles have different densities, and you can take advantage of another type of centrifugation, which is actually known as the equilibrium density gradient centrifugation. The equilibrium density gradient centrifugation is a characteristic feature for this type of centrifugation is that the centrifugation takes place through a density gradient from a low density material on the top to a higher density material at the bottom. One of the common material is sucrose, but we can just use cesium chloride, a very powerful gradient maker, or we can just use a phycol gradient. There are many different commercially available gradient makers, but sucrose is a very common one as well and readily available. And along with that, the purification scheme is that you take the organelle fraction of interest and you layer it on top of the gradient and apply your centrifugal force, and the organelle will move through. So you load the sample right here, and the organelle will move through right here, and they will actually arrive at the area where they are at equilibrium. So when lysosomes from the top reach the area of the gradient, which is 1.12 gram per centimeter per cubic centimeter, lysosome will actually stay there. Whereas see that mitochondria is still moving and it will stop at 1.18 G. Peroxisome still moving, it's going almost to the 1.23 G per cubic centimeter. So this example will end up with the lysosome at the top, then you have the mitochondria, and you have the peroxisomes at the very bottom. So how do you isolate these purified organelles from the gradient? So you actually puncture the bottom of the tube and you start collecting the fractions drop by drop. And we started from here, then here, then here, then here, and so on. So what happens, you collect set volume into different tubes. For example, you collect like 100 microliter in one tube, another 100 microliter in the second tube, and so on. Once you identify which tube has your organelle of interest, then you can actually conduct the biochemical and functional studies on your purified protein. So here, another technique, is also used, which is the purification of organelle by specific antibody. So here what we are looking at, that a vesicle has a protein which is called a clathrin. So you actually have the primary antibody against clathrin, and then the process is that when you actually have a cellular fractionation, in the cellular fractionation, you actually first add your primary antibody, which is an anti-clathrin antibody. So all the antibodies will bind to the clathrin. Now what you do, that in that sample, you actually add broken Staphylococcus aureus cell homogenate, or the cells which actually do contain protein A. Sometimes the protein A's are also available commercially, but suppose you just add the Staphylococcus aureus cells. These have protein A. And you know that protein A actually binds to the Fc region of the antibodies, and now all the vesicles are actually captured. You can just do a low spin centrifugation, and you can actually isolate all these vesicles. And once you actually isolate the vesicles, then you can disrupt this and you can have a purified vesicle. So here you can see that this is a transmission electron micrograph, and right here is the cell. On top of that, these are the coated vesicles that you can see through this transmission electron micrograph. So this is your specific antibody specific purification. So let's say that we are interested in determining all the proteins found in an organelle, and we just randomly pick up an organelle, say for an example, mitochondria. So every protein in the mitochondria you want to know. So you can do this by a mass spectrometry, and this diagram actually shows the mass spec. So how this actually works, so a typical mass spectrometry, there are many different types, is called MALDI-TOF, which is a matrix-assisted laser desorption ionization time of flight. Big mouthful name, but let's see that what is the principle. If you have the sample that you are trying to characterize, you absorb onto a metal target, pulse of a light from a laser will actually ionize the peptides in the mixture. You need to actually convert them to peptide. We're going to talk about that in the next slide. And then what you do, that once they become charged due to this incident laser, they will be actually ejected from here, and they will accelerate towards another detector. All the particles will move at a different rate. So if the particle is actually small right here, those particles will move faster compared to the particles which are much heavier. So what you actually do, that it takes time for the particles to move from their original position to the detector, is known as the time of flight from here to here, which is proportional to the square root of the mass of the particle over the charge. So again, this is called the m/z ratio, and that is how you can actually identify them, that which is first going in there. Lightest ion arrive first, and you can see their intensities as well. Molecular weights of each of the ions from the sample is calculated based on the time of flight. So now you can use to detect all the proteins in a purified sample. And let's take a look at how we do this. So let's say you have purified the mitochondria by differential centrifugation, and you are asking the questions, what proteins are found in this mitochondria? So after the differential and density centrifugation, purified mitochondria, you actually take all the proteins and digest it with a protease, say for example, trypsin. So if you do a trypsin digestion, pretty much all the lysine and the arginine sites, they will be cleaved, and you will have a bunch of peptides from all these proteins. And these we are calling as a tryptic peptide. And then you actually absorb those samples onto the specimen holder, shine the laser beam on top of that, and all these small peptides that you have produced will actually pick up the charge, and they will accelerate towards the detector, and the smaller ones will actually go faster compared to the larger particles. So each time the time of flight is detected, you will get a signal. So what happens, that you actually compare these flights, and then you can actually tell that the molecular mass of this. Now what happens, that the computational process is extremely robust. So for each of these, you can find a mass of a peptide. Now, if you actually do have the sequence of the proteins from the database. Almost all sequences of these are known. Say, for an example, if it is a human being, the human genome is already sequenced. So basically what you can have from here, that you actually have a mass, which is a protein sequence. And from that protein sequence, you can have all the possible nucleotide sequences, and that you can actually compare with the database, and the computer will churn these numbers. It's a huge task, but fairly quickly, to identify that each peptide is made up of which amino acid sequence. And it can tell you the exact amino acid sequence, assemble them together to that, and can tell you that what are the proteins that were present in there. A very excellent technique, and there are some modification of this technique, type of mass spectrometry that has been used with the bioinformatics proteomics to identify many different proteins. So that will be the end of the part 4, which is a chapter 4.4, isolation of the cell organelles, and that actually is the end of the module 1.1.